Good morning, everybody, and welcome to the first part of our lecture on imprinting technology. This fourth lecture of the course Experimentation in Science and Engineering, we will take a tour into a world that's very familiar for us. Us at the Sensor Engineering Department, we are very invested in doing biosensor research and imprinting technology research, and we use molecularly imprinted polymers in sensors to create solutions for everyday problems. And the reason we chose this case study is, of course, because we can make a meaningful contribution. We can tell you how research in this field actually works and how you can use that to solve problems in society and create a business case study around it. To explain how imprinting technology is used in sensor research, it's very important for us to start at the basics. We need to explain you what biosensors are and how imprinting technology can make imprinted polymers that can offer solution to traditionally shaped biosensors. If you look at the world around you, sensors are very important. We all have sensors in our own body. For instance, our eyes are able to capture light, and this can be light directly emitted from a lamp, but it can also be light that is reflected or diffracted from a certain object, and this allows us to see. Likewise, our ears are like satellites that capture variations in pressure in the air and allow us to capture sounds, gives us the ability to hear. However, when we think about sensors, we usually don't think about our senses. We think about a technological device. We think about a motion sensor, for instance, that detects our motion, because that's why it's a motion sensor, of course, and that drives an actuator, something that acts to open a door, for instance, so a mechanical action. Or whenever you enter a room, a motion sensor will detect that and switch on the lights. And that's usually how this technology works in real life. You have a sensor that detects a certain physical or chemical occurrence or an event and will translate that into an action, into an actuator. Another well-known example is, of course, a fingerprint scanner that you use in your passport to identify yourself or that you use to unlock your smartphone. Biosensors are a very specific subcategory of sensors. Biosensors don't detect a physical phenomenon usually. They usually detect a chemical entity. And in order to do so they use a bioreceptor. And this is, of course, what makes biosensors, the fact that they use a bioreceptor. Now, whenever a bioreceptor that is very specific for their target captures its target, we, as the end user, still needs to know that the target is captured by the receptor. So we need something that translates the capturing event into a measurable signal. And this is where a transducer kicks in. So a transducer will translate the binding event into, for instance, an electrical signal. And this we can read out using a computer, a laptop, but even a smartphone is possible. To get back to our first core element, the bioreceptor, there are four main categories of bioreceptors that are traditionally used in these biosensors. The first one being a nucleotide-based sensor, where you use either DNA or RNA on the sample on the surface of your chip. And what this DNA or RNA does is capture a complementary strain of DNA or RNA. Now, for instance, you can use DNA that contains a mutation, and then when a patient has a drop of blood added to the sensor and that has that mutation, it will bind to the sensor. If he doesn't have the mutation, it won't. And this way, you can use that type of DNA sensor to do research towards very rare diseases, hereditary diseases, or even cancer that contains mutations, of course. A more modern or more actual example of such a sensor would be an RNA sensor that is able to complementary bind to the RNA molecule that is the COVID virus. This way you could maybe detect COVID. Another class of bioreceptors that's often used in biosensors are enzymes. Enzymes are present in human but all animals' bodies. And what they do and what makes them so special is that they detect their target in a very specific way and then catalyzes the conversion of that target into another product. So it helps us metabolize our food, for instance. And in that process, molecules are released that you can then measure using your transducer. Similarly, you can isolate antibodies. from a living organism. And if you look at most fast tests that are now being developed for COVID, they are usually based on antibodies because antibodies in our body or in the body of mice are very specific. They recognize a very specific protein called an antigen uh, via a special region on that antigen called an epitope. So in case of COVID, this is usually the spike protein, and usually it's one or two or three amino acids on that spike protein that are specifically detected. And this makes your sensor very specific towards COVID. So your own DNA won't trigger a signal from the sensor, but a COVID particle will. So that's how these antibody or immunosensors actually work. Now, the fourth category is a little bit of a special category because it uses a more complex bioreceptor. It uses whole cells or whole organisms in a way that it uses, for instance, human cells or bacteria. And when you expose these cell-covered sensors to, for instance, a toxic gas, these cells will die. They release, for instance, a charged molecule, and this charged molecule we can measure using an electronic transducer, for instance, something like you use in the lab, a multimeter. This is where transducers kick in. Transducers, like we said before, will translate the binding event into a measurable signal. Now, the most or the oldest category and maybe the most popular category for this is optical sensing because it's so easy, right? Something binds to the sensor and we see an optical signal that we can see with our bare eyes. It can be fluorescence, it can be an optical um, color change that we can like see with our own eyes and then measure with a simple absorption meter like a camera on your smartphone. The second set of transducers that's also very popular are the electro or the electrochemical sensors, which are basically similar to the multimeters used in the Fitbit lab that just measure either voltage or current or resistance. The final category of sensors are what we call physical sensors, and there are two broad subcategories in this. The first one being thermal sensors, which are basically just fancy thermometers, and the other very popular category are acoustic wave devices, which are basically very sensitive scales, just balances like you use in the lab. And how they work specifically, we will explain later in this lecture. Now, to give you a brief overview of what you can use biosensors for in everyday life, I will start by giving you some very famous examples that are used everywhere in modern day society and that you might not even know that were biosensors to start with. Now, the most striking success story is the blood glucose meter for diabetics. And this blood glucose meter consists of a dipstick where you can add a drop of blood, and that dipstick contains two electrodes. And on one of those electrodes, there is an enzyme called glucose dehydrogenase. And that glucose dehydrogenase will catalyze the conversion of glucose into gluconic acid. And with that, uh, hydrogen ions, 2H plus in the scheme on the right hand side, will be released. Now, when you insert this dipstick into a device, a glucose meter. This is actually just basically a very, very basic multimeter. And what this multimeter does is apply an electrical field over the two electrodes. So when the hydrogen atoms are released, they will travel inside the electrical field and make a current, because that is what current is, right? Ions moving in an electrical field. And the more glucose is present, the more current will be generated and the bigger the signal is. So your device will give a higher signal, and this can be calibrated to give the end user not a signal, not a measure of how much amps is flowing, but actually the concentration of glucose that is there. And this has changed the life of diabetics dramatically, because before they had no real measure of how much glucose was in their blood. And this is very important for diabetics, because when the glucose concentration is too low, you need to eat, right? Because otherwise you can faint. When the blood glucose concentration becomes too high, you can die, so you need to insert insulin. And before the blood glucose meter, the only way to know what the glucose concentration in your blood was basically to go to the hospital, have a blood test, and 2 days later you knew, which is obviously much too late. Uh, with this invention, diabetics were able to measure their blood glucose levels at home, and if required, they would then eat something to raise their blood glucose levels, or they would take an insulin shot to bring the glucose levels back down again and save them from getting a stroke or passing out. Another very important example of biosensors and a success story of biosensors that you might not have even known that were biosensors is the home pregnancy test. And a home pregnancy test, I mean, the word says it itself, uh, indicates if you are pregnant or not. And the reason that it's so important is one, it's a very commercial success story. You can buy these tests in nearly every supermarket. And two, it's based on lateral flow assays. And this is very important in biosensor research because lateral flow assays are very popular. Now, a lateral flow assay is actually just a glass slide that contains multiple pads. You have a sample pad where you apply, well, your sample, in this case, a drop of urine, and then through capillary forces, the drop will on its own move to a second pad called the conjugate pad. And there, on that conjugate pad, are antibodies coupled to gold nanoparticles. And these antibodies recognize a hormone in the urine called HCG, human chorionic gonadotropin. And this HCG is only present in the urine when you're pregnant. So what happens is when the hormone is present, these antibodies will bind to the hormone. These this configuration, so the analyte plus the antibody will then travel to the test line, where there's a second antibody that will also recognize the HCG, and this will bind to it. And because the gold conjugates are attached to the other side, these gold conjugates will give a color, and this you will observe as a stripe. On a second test line, there are also antibodies, but these antibodies on the control line are not focused onto the hormone, but onto the antibodies with the conjugates itself. So they will always bind the antibodies that are not bound to a conjugate. This means that when you're not pregnant, all antibodies will bind to the control line and give a solid line there. When you are pregnant, only some of them will bind to the control line and the rest will bind to the test line. Now, when the test line is apparent and the control line, you know that the sensor still works and you know that you're pregnant. When only the control line works, you know that you're not pregnant. And that's how the sensor works. And this has obviously turned into a huge commercial success story because, like I said, like you can buy these tests in nearly every store. They are very elegant and it takes you about a minute to know if you're pregnant or not, which is a huge difference with going to the doctor's office and letting them check your blood and having the results a couple of days later. So this has changed again the life of many people. A more modern example or more technologically advanced example that I quickly wanted to show you is an example that's developed by a partner of us called Unisensor in a project. And they work a lot on food safety. So what they wanted to do is they wanted to make a sensor that is able to detect a contamination in a food product, in their case, antibiotics in milk, because that's a huge problem in the milk industry that they give antibiotics to cows. This ends up in the milk and this ends up into the consumer's system. And you don't want that, of course. So what they did is they had a gold chip which they coated with antibodies for antibiotics. And this they connected to the prism. And the reason they did this is that in this way you can use it in combination with a transducer technique we call SPR. And we're not going to go into detail about SPR because SPR is a difficult technique to explain and would be technologically probably too advanced for this course. But the reason I wanted to show you to it nevertheless is to show you that optical devices are sometimes more complex than just a color reaction or a fluorescent signal. What happens here is you have a light source, which is a laser, that reflects off the prism and then falls into a detector array. These are all small cameras that can detect the laser. Now what happens when a substrate binds to the antibodies is that the laser will still be diffracted, but at a different angle. So a different camera will capture the signal. And the more difference there is, the more targets, so the more antibiotic will be present in the sample. This is how this sensor works, and this is commercially sold to many food producers within the European Union, but even outside the European Union. So it's also turning in quite a success story. Now we talked about the advantages of using bioreceptors for sensing. They're very specific. They're usually biological, so they're chemically active, so you can couple them to a substrate. But they also have drawbacks associated with them. Normally operate within a human body where the temperature, the pH, the concentration of certain ions are all very controlled. Now, this is obviously not the case when you take them outside these living organisms, so they're not very stable. So you need to create a situation where they are stable outside the human body. This makes it, of course, more expensive. And also, in the long run, they won't have an unlimited shelf life. They have an expiry date. So these are disadvantages, obviously, when you want to use them in a commercial setting. On top of that, they're made in transgenic animals. Usually, you immunize a mouse with a certain antigen, and you make the mouse make antibodies, and you harvest these antibodies, which is a time-consuming and a costly process, and also not very ethical, right? Because you're basically sacrificing the life of mice and rats to create a sensor. Now, that's why they invented a new technology called recombinant reactor technology that is able to do the job faster and fully synthetically, so no animals involved. However, this is even more expensive because these reactors are very, very complex structures and they cost a lot of money. So that is another drawback. So we need something that is more stable than these receptors, and we need something that's easier and cheaper to produce. On top of that, not every target has a natural receptor, right? Sometimes there are no antibodies or enzyme towards, for instance, when you want to detect a toxic substance. You can also not make antibodies in mice because when you insert toxic substance into the mouse will die and will not make antibodies. So these are problems that are drawbacks of using bioreceptors and sensors. And here, imprinting technology can offer a solution. And how that works is something which I will explain in the next part of this lecture.